So circulating tumor cells, why do we study them? Essentially in the carcinoma world are ahead of us. They've done a lot of work and they've shown that just counting them you can correlate with disease burden and prognosis, and actually further characterization of the CTCs can give information about understanding processes of metastatic disease. And of course this is relevant to osteosarcoma because it will metastasize particularly through the bloodstream. So this is how an orthopedic surgeon thinks about how osteosarcoma occurs. And as Prof. Flanagan was saying, there's some catastrophic effect in probably an osteoblastic cell, you may agree, but we don't know what the exact effect of that is, becomes an osteosarcoma cell. And my hypothesis, I've been working on this for a good 10 years, is that the osteosarcoma cell will be overexpressing MT1-MMP. This is a membrane-bound matrix metalloproteinase that cleaves type 1 collagen in bone, will allow the osteosarcoma cell to destroy its own bone, and also the MT1-MMP can activate other MMPs, such as MMP2 here, which will cleave other proteins and essentially allow the cell to escape into a blood vessel and metastasize. So now we're thinking about CTCs and we're thinking about the sarcoma cell in the blood vessel expressing our marker. The other marker we thought would be better to have more than one marker, and we came up with NG2. That was from the literature, and also Donald Salter, who's a pathologist in Edinburgh, has studied this, I think, with a BCRT-funded grant, and showed expression. So in our cell lines, it has been a useful marker. But of course, identifying your circulating tumor cells is quite difficult because in a blood sample there'll be lots of white blood cells, even more red blood cells, and you may only have a handful of your cells of interest in your sample. Anyway, we got going with some pump priming fundings from a local charity, the Just Say Yes Fund. The BCRT came through with some pump priming funding as well. And basically what we did was confirmed our markers by spiking cells into healthy volunteer blood. We were able to show the orange peaks here are our osteosarcoma cell lines spiked in, and they're shifting away with our MT1 and NG2 markers. From the different peaks here are the different types of white cells in the blood which are relatively negative. So this was working quite well, and so we can have a basic system here where we're spiking cells in, and you've got the residual dots here are white blood cells. So this actual first plot, there's no cells spiked in, 6 false positives. Loads of cells spiked in, 100,000 cells, you've got 85% recovery. And reduce the numbers of cells spiked in. The recovery varies a bit, but this is what we got to. It's reasonable recovery. So with a quick flow cytometry assay, we're actually getting somewhere. And so we went for more funding to set up a study in patients, and Children with Cancer UK came up with funding, and a local funder, JGWP, funded us as well. And we were able to set up a UK-wide study. So I wrote the ethics to recruit children and adults with osteosarcoma. So that ran for just over 3 years, ended July last year, and did quite well actually. The recruitment got to our initial target of 30, so I got a bit cocky and thought we'll go for 50, and recruitment stopped for a few months. But we did get to 45, which I think is a reasonable achievement actually of getting the samples. And we had 4 centres open, Edinburgh, 4 patients. We recruited 12 at Newcastle, Birmingham Children's, 4 in London at UCL, recruited really well, 25 patients. So in terms of the results, total of 45 patients recruited. So our basic, what I call the sort of low resolution flow cytometry assay is very quick. You can get the sample and have it analyzed and be looking at the data 3 hours later. The useful data, we did have to change the MT1 antibody when we were using patient samples. The initial antibody wasn't so clear, but we started to identify clearer populations after changing the antibody. So we've got 18 useful plots to analyze from patients, and actually 8 patients also serially sampled. And we managed to do some extra things as well towards the end of the project. So this is just to show, so we basically, it's a bit complicated, but basically we were looking at our MT1 and NG2 positive areas. So we split these into quadrants, which we always defined up here just above 10 to the 3 and 10 to 2.5 here. But basically, if you add these quadrants up in a healthy volunteer, it's always less than 5%. cells spiked into healthy volunteer blood, an example here at 21%, and patient 15 from UCL coming up at 14%. So quite an interesting discrete population. Post-treatment disappearing. So this is what we started to see. And actually, if you put it together, and we did a forest plot, and most of these are 18 osteosarcoma patients. There's a few soft tissue sarcoma patients in there as well. The blue squares are osteosarcoma patients. And actually looking that sort of combined percentage there, it actually came to statistical significance at diagnosis. So that was our first result when we analyzed the data. And also serially sampling patients. Here's patient 10 from UCL with a 14%, and at follow-up, an increasing percentage. A bit worrying. I need to feed this data back to see if it has this patient relapse or has got disease burden. This is a patient of mine from Newcastle who had, in follow-up samples, an increasing percentage. Here he didn't have Frank Metz. Here he's got Frank Metz and increased percentage. Here's a series of UCL patients, some with quite alarming percentages, dropped off at follow-up samples, and I'd hope they're in remission, so we'll feed that data back and see if we're right. So in summary, CTCs and osteosarcoma, we can count them with a quick flow cytometry assay. More work is required to assess whether it will hold up really in the long term, and of course it's going to be used in ICONIC for further assessment of this. And I'll just show you a bit more detail when we had enough of a blood sampled. We're doing some high resolution flow cytometry for more physical characteristics and some sequencing. Claire Coyley was a patient with osteosarcoma I looked after in 2002, passed away. Her mother kept fundraising and gave us some money to do some further work. So when bloods are coming in, this is the quick assay, but also any remaining blood we would use it for high resolution flow cytometry. And that basically is a flow cytometer. It's quite labor intensive, but it will give you a picture. It's got a camera as well as a dot plot. But of course you get it's much nicer, you get all these nice pictures. If you put a cell line through, positive for MT1 and NG2, for example, so osteosarcoma cell lines, and if you spike them in, that's a white blood cell, positive for CD45, for example, and then you see our spiked cells there. Patients, you get all sorts of interesting pictures. You've got clusters, you've got large cells, small cells, you've got white cell clusters, but some evidence that you've got CTCs here, that, given that they're CD45 negative. The next thing really is to try and get further proof from sequencing. We had an interesting sample came up from Birmingham as a follow-up patient. I suspect this, I don't know yet, but I think they sent it because perhaps the patient had relapsed. We actually put this through the Parsortix. You'll hear about that from Sheffield shortly, just to enrich the sample, interesting pictures again, and we split the sample, so some for DNA extraction and amplification. We did some PCR to check with a housekeeping gene, we get nice curves. Sent it for TP53 sequencing, but unfortunately we sent 2 patient samples. We got intronic deletions only, didn't really tell us a lot, but at least we could get DNA out of these cells for sequencing. We've sent that off for exome sequencing now. So that's as far as we got, and we'll keep working on it in the ICONIC study. So acknowledgements are really to the technicians and postdocs and students who've worked on it, and BCRT, Children with Cancer UK, JGWP, who funded this study. I'll just go on very quickly to about imaging improvements for osteosarcoma surgery. I think Craig already set the scene for this. The problem we have, and this is an example of a patient who presented to me a year ago, osteosarcoma in the proximal tibia. That's obviously in the bone. It's bursting out the back into the soft tissues. So that's the definite tumor on the scan. We're worried about this area here, and it's close to the main artery, and the surgeons in the room will know this is always the dilemma about should we amputate or save the leg. The patient was really very reluctant for amputation, as a lot of them are, so we did a limb salvage and a proximal tibial replacement, and actually the back was fine. What was a bit of a surprise was the closest margin was up here by the patella tendon, and I don't think the imaging could tell us that really. That was quite a surprise. And of course, necrosis down at 60%, and we know from the Birmingham classification that's not a good situation. So we really need better options in order to enhance our MRI actually, and there are some options that I'll show you coming forward. Interestingly, when you're correlating later, I think this was tumor here and this was just edema actually when we started to look at it more closely with, of course, with the pathology result. we have been running a sarco PET study up in Newcastle. This is MRI PET for patients with soft tissue sarcoma, and actually myxofibrosarcoma in particular, because these are quite infiltrative, and often we're not sure where to go in terms of how wide you want to go. This is a patient with a myxofibrosarcoma in the chest wall. It has two components, a sort of lighter grey in the sequence and a darker grey. I'm not sure anyone could tell you, even the radiologists in the room, I'm not sure they would say what that means, but actually the patient then was recruited to the study and had a PET scan with his MRI, and actually that darker area is very, very avid. So he had a surgery planned off the standard MRI that showed a deep margin, and therefore he had to come back, a positive deep margin, so he had to come back and have a formal rib resection. And so really this is the principle that could be applied back to our osteosarcoma. I fully appreciate the fact that a post-chemo scan with necrosis on FDG PET might not give us enough information to distinguish what's necrosis and what's avid tumor, but I think we have to start somewhere. The other point is about intraoperative imaging, and this is Craig training me a few years ago. You know, we're sort of operating blind, you know, as a surgeon, it's quite uncomfortable. We're often trying to resect a sarcoma and worried about how close we're getting to it. And so we really need to move forward. Fluorescence guidance is already into the clinic, again, in the carcinoma world. The principles are you can do non-targeted or targeted image guidance. And this is colorectal surgeons in Leiden. They're looking for liver mets. So on the liver surface there, the photograph, you can't see anything much, but the patient has been injected with a monoclonal antibody to EGFR with a fluorophore, this indocyanine green attached to it, injected a week before, and actually when they turn the fluorescence light on, the metastases light up. And this is really transferable to the sarcoma world. We're already looking to do our first soft tissue sarcoma patient with a non-targeted, just using the dye alone, and if the sarcoma is vascular enough, we think it may light up. We've got the dye, we've got a fluorescence camera from Stryker that we're going to try and use. But if we're talking about sarcoma targets, we've got targets identified: MT1-MMP, NG2, CD44, death receptor 5. We've got antibodies working up on this for our paraffin tissues to identify suitable patients. We can conjugate radiotracers and fluorophores to these antibodies that need to be humanized before they go into the clinic, but there is a work program there. If we look at MT1-MMP, I already know that in a series we've got 60 osteosarcomas, high grade. Over 70% of these overexpress MT1-MMP. This is a target that's already got in a clinical trial at the Marsden for carcinoma and sarcoma, actually. But high-grade sarcomas overexpress MT1-MMP as an example there. And we have the workflow where we actually grow our cell lines in bulk. We've got our positive and negative controls, and we can take the tissue from the archive and put it on the slide. So you can see something like this, where we've got your MT1-MMP overexpression, your MCF7s are negative. And then this is actually a myxofibrosarcoma invading into an adjacent lymph node. You can see very clearly this is the margin we're worried about when we're operating. And if we could target that preoperatively and intraoperatively to see the active margin, that's really the principle. So already underway in Newcastle, we have a radiochemist putting zirconium probes onto our monoclonal antibodies after addition of a radioreceptive ligand, so we can do some in vitro radio stability checks. But we need funding to go on to do, we're getting a SPECT CT, so we can do small animal PET, and we do have a working mouse model for this. And same principle really, if we're going to do intraoperative fluorescence guided surgery, targeted, so again, binding your fluorophore onto your monoclonal antibody using a fluorescence camera and correlating with histology. So that's it. In summary, I think image guidance for osteosarcoma surgery has the potential to improve our decision-making preoperatively. Down the line, it might start indicating chemo response before we do the surgery. So I think it's worthy of looking at funding this type of work. And it would be called sarcocyte. There you go.